Glycolysis. So the word glycolysis is derived from two Greek words, glykos and lysis. Glykos means sugar and lysis means splitting. The splitting of the glucose occur, a reaction through which the splitting of the glucose occur. So that is glycolysis. Okay. So this reaction occurs in the cytoplasm, inside the cytoplasm in all the living organisms. The splitting of the glucose occur during respiration. In all the aerobic organism and anaerobic organism, this glycolysis will be always there. Okay. So through this glycolysis, what will happen? The 6-carbon sugar get converted into 2 3-carbon compounds. The 6-carbon compound split into 2 3-carbon compounds known as pyruvic acid. So these 2 pyruvic acid molecules are produced from glucose, okay, through this process. Okay. So during this process, a lot of energy, some amount of energy from the glucose is released. So that energy is stored in the ATP molecules and NADH2. So the released energy get stored in the energy-rich compounds like ATP and NADH2. Okay. During this process, 2 ATP and 2 NADH2 molecules are produced. Okay. So this is the overall process. So it occurs through a series of 10-step enzymatic reaction. The glycolysis is not a single step conversion, it occurs through 10 steps overall. Okay, thus the reaction completes. Okay, so we can go through the process. This glycolysis reaction, it was discovered by 3 scientists, Gustav Embden, Otto Meyerhof and J. Parnas. Okay. So we also be termed it as EMP pathway. So this glycolysis can also be termed as EMP pathway. So Embden, Meyerhof and Parnas pathway. Okay. So the inside the cytoplasm, the type of carbohydrate that is mainly available is the sucrose. Sucrose is the main source of carbohydrate inside the cytoplasm, especially in plants. Okay, so that sucrose is a disaccharide, so it need to be converted into glucose and fructose. Need to be broken down to glucose and fructose. Then only the glycolysis can start. Okay, so for that purpose, in plants, the sucrose, the enzyme invertase break down the sucrose into glucose and fructose. So here from glucose onwards the reaction starts. Okay, the glucose first converted into glucose 6-phosphate with the help of the 1 ATP molecule. So that means 1 phosphorus is added to glucose. 1 phosphorus is added to glucose. We know that glucose is a 6-carbon compound, so phosphorus is added to the 6th carbon from ATP. From ATP donate the phosphorus, so that it become 6-phosphate. 6th carbon, there is a phosphorus, so glucose 6-phosphate will be produced. So with the help of the enzyme hexokinase enzyme, that enzyme is very important, hexokinase enzyme. Okay, then in the next step the glucose 6-phosphate get converted into fructose 6-phosphate with the help of the enzyme hexose isomerase kinase. The glucose 6-phosphate get converted to fructose 6-phosphate with the help of the enzyme phosphohexose isomerase. So that is the enzyme for that process. Okay, so that is a simple conversion, converted into fructose 6-phosphate. So that is an isomer. Fructose 6-phosphate is an isomer of glucose 6-phosphate. Okay, in the third step, what will happen? The fructose 6-phosphate. In the third step, the fructose In the third step, one ATP is added to the in the third step, one phosphorus is added to fructose 6-phosphate. One phosphorus is added to fructose 6-phosphate. One phosphorus is added, so it becomes fructose 1,6-bisphosphate, like this. So here one more phosphorus is added here. So one first carbon and the sixth carbon phosphorus is added. So thus it become fructose 1,6-bisphosphate. 2 phosphorus added. So here again one more ATP is utilized. Okay, here the enzyme is phosphofructokinase. Phosphofructokinase enzyme. So that enzyme is important, but in NCERT that enzyme is not mentioned. So for need purpose, not that much important. The phosphofructokinase enzyme. Okay, so for glycolysis this enzyme is important. So this step is known as rate limiting step of glycolysis, this conversion. Okay, that means whenever the concentration of ATP increases inside the cell, whenever the concentration of ATP increases inside the cell, this enzyme get inactivated. That means when there is lot of energy inside the cell, this enzyme get inactivated. So this conversion never happen. This conversion we can term it as rate limiting fructose 6-phosphate to fructose 1,6-phosphate. So is controlled by the enzyme phosphofructokinase enzyme. Okay, the this enzyme can be get activated when there is not sufficient energy inside the cell. When there is no ATP, suppose in the cell doesn't have sufficient energy. So if there is a deficiency of ATP inside the cell, this enzyme get activated and the glycolysis will continue. So that through the glycolysis the cell can produce more ATP. Okay, so if there is a lot of ATP, if there is a large amount of ATP inside the cell, what will happen? The enzyme inactivated. This enzyme, the activity of this enzyme retards, so that that conversion will not occur or cannot occur. So that means the cell doesn't need energy now. So there is no need of breakdown of glucose. There is no need of ATP. So that this reaction can be almost stopped here, or otherwise we can say the reaction will be very slow, this conversion. Okay, so so that means according to the requirement of the cell, it can speed up or slow down the metabolic in this metabolic pathway. It can speed up when there is requirement of ATP. Okay, so if there is no need of ATP, this reaction will slow down. So who is responsible for that? This enzyme. This enzyme can do that process. Okay, so that is why this step we can term it as the rate limiting step of glycolysis. Rate limiting step of glycolysis. Speed controlling step of glycolysis. Speed of glycolysis can be regulated with the help of this enzyme. So okay, so that enzyme can also be termed the pacemaker enzyme, speed controlling enzyme, pacemaker enzyme. So that is the third step. Okay, then in the next step, the fructose 1,6-bisphosphate. So that is the 6-carbon compound broken down into 2,3 carbon compound. Okay, so that suppose this is the 6-carbon compound, we know that. So it split into 2,3 carbon compound in the next step. So that is what happening here. So here the two compounds are 3-phosphoglyceraldehyde and dihydroxyacetone phosphate. So these are the 2 3-carbon molecules, compounds produced here. Okay, by the enzyme aldolase. So here the enzyme involved is aldolase. Okay, So here these compounds can also be termed as triose phosphate. Okay, these two molecules both are triose phosphate. Okay, so that means 3 carbon sugar or 3 carbon compounds. Okay, so here among these two molecules only the phosphoglyceraldehyde can continue the reaction. The reaction can continue from with the help of this 3-phosphoglyceraldehyde only. Okay, so here 1,3-phosphoglyceraldehyde is here. Then what will happen? The dihydroxyacetone phosphate again converted into 3-phosphoglyceraldehyde. Okay, with the help of another enzyme. Okay, so that conversion will occur. So that is the fourth or fifth step of glycolysis. Fifth step of glycolysis. So this is the fourth step, and this is the fifth step of glycolysis. So here as a result, what we can see here, there are 2 molecules of 3-phosphoglyceraldehyde. Okay, 1,3-phosphoglyceraldehyde is produced from dihydroxyacetone phosphate. So we can see there are 2 molecules of 3-phosphoglyceraldehyde. Okay, here then this molecule 3-phosphoglyceraldehyde then continue the process. Glyceraldehyde then converted into 1,3-bisphosphoglyceric acid. Converted to 1,3-bisphosphoglyceric acid. So there occur an oxidation. That means 3-phosphoglyceraldehyde release hydrogen ions and electrons, remove hydrogen ions. Okay, so when 1,3-phosphoglyceraldehyde get converted to 1,3-bisphosphoglyceric acid, 1 hydrogen ion and 1 electron is removed. Okay, the 3-phosphoglyceraldehyde then convert into 1,3-bisphosphoglyceric acid. Okay, during that process hydrogen ions are removed from 3-phosphoglyceraldehyde. Hydrogen ions and electrons are removed from 3-phosphoglyceraldehyde. So these are again received by NAD molecule. 1 NAD receive 2 hydrogen ions and 2 electrons. Thus it get converted into NADH2. NADH plus H plus NADH2. Okay, so that is the conversion. 1,3-phosphoglyceraldehyde when converted into 1,3-bisphosphoglyceric acid, 1 NADH2 will be produced. 1 NADH2 will be produced. So here I already told you there are 2 3-phosphoglyceraldehyde molecules. 3-phosphoglyceraldehyde molecules. There are 2 3-phosphoglyceraldehyde molecules. So we can say 2 NADH2 molecule can be produced. Okay, 2 NADH2 molecule can be produced from 2 NAD. Okay, so that is the process. So during that conversion, another thing is that 1 phosphorus is added to the triose phosphate. 1 phosphorus. So thus it becomes 1,3-bisphosphoglyceric acid. 1,3-bisphosphoglyceric acid. 2 phosphorus is there. You can see in that 3 carbon compound. 2 phosphorus is there. So here we can see. So there is no involvement of ATP. ATP is not participating in this reaction. Okay, ATP is not participating in this process. But the phosphorus came from the cytoplasm only. Only the inorganic phosphate, some inorganic phosphate is added to the triose phosphate or 3-phosphoglyceraldehyde. Okay. Then, so here the 1,3-bisphosphoglyceric acid now become 3-phosphoglyceric acid. Okay, so that means 1 phosphorus is removed from 1,3-bisphosphoglyceric acid. Okay, so that phosphorus is added to ATP. Okay, added to ADP. The removed phosphorus is added to ADP. Thus ADP get converted to ATP. So when 1 molecule of 1,3-bisphosphoglyceric acid get converted to 1 molecule of 3-phosphoglyceric acid, 1 ATP will be produced. Here we know that there are 2 molecules of 1,3-bis phosphoglyceric acid. Okay, and it get converted into 2 molecules of 1,3-bis phosphoglyceric acid get converted to 2 molecules of 3-phosphoglyceric acid. So that means 2 ATP molecules are produced. 2 ATP molecules are produced during this process. Okay, so this is an energy yielding step of glycolysis. So this is here from where that phosphorus came, from the substrate molecule. From the substrate, the phosphorus is given to ADP. So that ATP synthesis can also be termed substrate level phosphorylation. From the substrate, the phosphorus is donated to ADP. Okay. The 3-phosphoglyceric acid again converted to 2-phosphoglyceric acid. So here the enzyme is there, phosphoglyceromutase. That is not important for you or the NEET level. So then 2-phosphoglyceric acid become 2-phosphoenol pyruvic acid. So here along with this, a molecule of water is removed. A molecule of water is also removed from this molecule. So we can term it as dehydration reaction. So here this is the only step in glycolysis where water is removed, dehydration occurs. So that is the conversion of 2-phosphoglyceric acid to 2-phosphoenol pyruvic acid. So here the enzyme is enolase. Enolase is the enzyme. In the last step, then what will happen? 2-phosphoenol pyruvic acid get converted to pyruvic acid. So here you know that each molecule occur in 2 times. 3-phosphoglyceric acid 2 molecule, 2-phosphoenol pyruvic acid 2 molecule, again that 2 molecules of 2-phosphoenol pyruvic acid again converted to pyruvic acid. Here again 2 ATP are produced. Okay, 2 ADP get converted to 2 ATP. So here the enzyme is pyruvate kinase. That enzyme is also important. Pyruvate kinase enzyme. Here the end product is pyruvic acid. How many molecule? 2 molecule. So that is the end of the process. So here at the end of the process we can say there are 4 ATP molecules produced. Here in this step 2 ATPs are there. Okay, here again 2 ATP. So the total number of ATP production 4 ATP. Then here we can see at the beginning we already studied here the first step and third step, ATP 1 ATP each utilized. Here 1 ATP here. So total consumed 2. So the net gain is 2 ATP. Net gain is 2 ATP, and in addition to that 2 NADH2 molecules are also produced. 2 NADH2. Okay, 2 NADH2 molecules are there. Okay, 1 NADH2 equal to 3 ATP. Okay, 1 NADH2 means 2 ATP. So 2 into 3, that is 6 ATP can be produced. Here 2 ATP. So the total is 8 ATP. This is the net gain of ATP in glycolysis. Net gain of energy. So net gain of ATP means totally directly 2 ATPs are produced. Net gain, direct gain, net gain 2 ATP, and 2 NADH2 molecules are also produced. That can be converted to 6 ATP. This 2 NADH2 can carry energy equal to 6 ATP. So thus we can say total energy obtained is 8 ATP. That means 2 ATP and 2 NADH2. Okay, so that is the overall process. So this is the 10-step enzymatic reaction. Okay, so here we can see what are the 10 steps. I already just mentioned earlier. So the first step where glucose get converted to glucose 6-phosphate. So this is the conversion. Second step, glucose 6-phosphate to fructose 6-phosphate. Third step, fructose 6 to phosphate to fructose 1,6-bisphosphate. Fourth one, fructose 1,6-phosphate to bisphosphate to and the splitting reaction, that is the splitting reaction. Fructose 1,6-bisphosphate split into 2,3 carbon compounds. So that is the fourth step. Splitting process is the fourth step, splitting process. Then again the dihydroxyacetone phosphate get converted to glyceraldehyde 3-phosphate, fifth step. Okay, sixth step means glyceraldehyde 3-phosphate get converted to 1,3-bisphosphoglyceric acid or glycerate, like that. Seventh step, 1,3-bisphosphoglyceric acid get converted to 3-phosphoglyceric acid. Then 8th, 9th and 10th. So these are the 10 steps of glycolysis. The end product means 2-pyruvic acid and 2-NADH2 and 2-ATP. So these are the end products of glycolysis. Okay, right.